Hello, everyone, and thank you so much for joining us tonight. It's a pleasure to be here with you. And it is a great honor of mine to introduce Professor Chris Love and his student Neil Dalvey. Chris Love is a professor in the Department of Chemical Engineering, where he holds the Raymond and Helen Saint-Laurent Chair. He received his doctorate in chemistry from Harvard under George Whitesides, and he did a postdoc at Harvard Medical School before joining MIT in 2007. Chris is working with his students to advance technologies with the potential to broaden access to a medicine at a global scale, ensuring that life-saving treatments are available and accessible and affordable to everyone who needs them. In 2007, he was named MIT's lead investigator on a multi-university grand challenge sponsored by the Gates Foundation to create a manufacturing platform that can produce vaccines at ultra-low cost. Tonight, Chris will be talking about how they applied this research to the problem of COVID-19. He is also joined by Neil Dalvey, a PhD student in his lab. Neil's research uses genetic engineering to create new yeast strains as an alternative host for biologic manufacturing. And my name is Heather Upshaw. I'm the development officer for chemical engineering. And I just want to say that it's been a complete privilege to support Chris and watch the progress that he and Neil, along with the researchers in their lab, have made at this critical moment. Please join me in welcoming them.
Good evening, good morning, good afternoon, wherever you may be calling from today. We're excited to be here with you today. I'd like to use this time, we're going to talk a little bit about how we're thinking about addressing some of the inequities that exist as we think about the access to medicines and vaccines, particularly in the context of COVID today. It's been an interesting year for all of us, and I think as we get started here, I just wanted to kind of share a couple of thoughts about where we are presently with the kind of addressing this current pandemic, as well as thinking about kind of what is next. So I think we should start with just remarking on the fact that it is amazing that in roughly a year's time, we were just reflecting that it's been almost a year since the campus kind of dispersed as a result of COVID, that we have 173 million individuals who've been vaccinated with new vaccines. And this is unprecedented, unheard of in the context of developing new interventions of this type. So this is just amazing. And for those of you that may have been involved in aspects of this, thank you for what you've done in that regard. At the same time, we have about 130 countries presently for which there's no vaccines that have been administered yet. And if you're reading the news, you understand that there are new variants that are emerging of the virus that may present certain challenges to interventions like some of the monoclonal antibody therapies that have emerged, as well as some of the vaccines. We don't know yet what the implications for this are going to be. So as we think ahead then, where do we still need to go? What still needs to happen? Well, if we kind of make some estimates about global populations, herd immunity, what's necessary to reach kind of protection globally, we're probably looking at on the order of 8.2 billion doses that are needed. And the price for those doses to be able to reach all parts of the world really need to be about $3 or less. Today in the US, the current prices of the vaccines that are being purchased is somewhere between $20 and $30 a dose. So we have an order of magnitude to go. And we don't know yet, as a result of some of the evolution of the virus, the transition of this from a pandemic to an endemic, what that will mean in terms of the number of boosters or the types of annual updates we might have to have for vaccines of this type. Is this going to be like the influenza, or is it more like pertussis or some of the early childhood vaccinations that you get maybe once or twice during your life? What we do know is that we're going to need more vaccines, and we're going to need innovations in manufacturing to reach that. It's not hard to read the news today to see that manufacturing is very much at the forefront of the conversations of how do we make these vaccines available. And what we'd like to do before telling you a little bit about some of the work that we're actually doing right now is to take you back a little bit to last year. Around this time last year, there was this interesting convergence of a number of things that were happening for us as a lab. As Heather mentioned, we've been thinking a lot about the ability to bring medicines to patients through innovations in manufacturing technologies. Today, many vaccines or other products of this type, biopharmaceuticals, are made in mammalian cells. They're made in large factories that are centralized in single locations around the world. We've been thinking about small automated manufacturing modular technologies that are more like like a desktop printer or refrigerator in their size that can make drugs from start to finish in an automated fashion using yeast that are more similar to those that are used to brew beer than are those that are used to make medicines today. These are some of the kinds of technologies that we've been thinking deeply about through research with the Department of Defense and the Gates Foundation to address the question of how do we make vaccines more affordable, and how do we use kind of new technologies to change the paradigm of centralized manufacturing to distributed ones. The convergence of all of this happened for us last year. As we were kicking off a new phase of our program with the Gates Foundation, they called and said, you know, guys, there's this problem. Turns out we didn't really understand how big that problem was going to be. And so I'm going to take you back now to where we started last year, and I'm going to hand it over to Neil, who was at the forefront of where we were when this new problem emerged.
Thanks, Chris. So I'll kind of tell a little bit about what this was like on the ground in the lab starting last February, really. So we had worked with, like Chris said, with the Gates Foundation on a project a couple of years ago, engineering a rotavirus vaccine to improve its manufacturing process, which is important to increase global accessibility to vaccines for rotavirus. And like Chris said, we use this yeast host that is kind of the main technology of the lab that is useful because you can grow yeast very cheaply. They just grow in sugar, as if, just like many of you are probably doing your sourdough starters around the same time. And another advantage of that system is that we can create yeast strains really quickly. So we can test vaccine concepts and make engineering changes to vaccines at kind of breakneck speed in a matter of weeks. So we had done this for a rotavirus project previously, and around February, when sequences to this new virus started to become available, people asked us, hey, can you check this out and see if you could make something useful as a vaccine component? And I think the numbers are that stories written about how fast Moderna went with their mRNA vaccine, and they went from sequence to having some sort of preclinical drug product ready to be tested in animals in about 3 weeks. And we did it in about 4. And those 4 weeks happened right as MIT was shutting down, and we started to have interesting data on potentially vaccine components that could be interesting. So Chris got exceptions for 4 of us to continue working in the lab. And so we kind of hit the ground running, focused on only this on truly an empty campus. And within 4 weeks, we had a preclinical product that we could test in mice. And within a month or 2, we had a product that was ready to be transferred to a real manufacturer, which we were connected with through the Gates Foundation to a large-scale vaccine manufacturer in India. And we literally took a yeast strain, froze it down, put it on a plane to India, which was in and of itself a challenge to find a way out of this country and into another country during the depth of all the lockdowns. And that first vaccine candidate since then has been manufactured at kind of the largest scale that you would manufacture something at for global access, and is currently in a phase 2 clinical trial in Australia, which is very exciting. So after having all this success early on, we were fortunate to be able to kind of then let the vaccine manufacturer handle that, and then we can turn around and get back into the lab and focus on improved vaccine candidates that we've shown to improve the manufacturability of these different proteins, improve the efficacy of vaccines, and that's kind of where we are today. And there's been no shortage of hot deadlines coming in with new variants and new findings at other labs. So as fast as we can pull information off of literally Wikipedia and Twitter, we can turn around and implement those sequences and concepts into new vaccine designs. So here we can just highlight a couple of the different tools that we use and achievements we've been able to reach over the last year since kind of turning our attention back into the lab on improved designs. And those include improved manufacturability of vaccines. So can we take this first concept that we started with and come up with something that is even easier to produce, that we could produce at lower costs and thus reach more and more people? And also, can we improve the efficacy of the vaccine? And so benefits of that could be, can we use a smaller amount per dose and spare more doses that way? Or could we have a vaccine that just requires 1 dose instead of 2 doses? So these are the kind of ideas that we're looking at moving forward.
I think this is kind of at the heart of what chemical engineering kind of brings to this question, is how do we take what's effectively a straightforward manufacturing and bring it to the largest possible volumes, and to do so with efficiency. And, I think as we've walked through this experience in the last year, we've learned a lot about the challenges of the molecules that are in the clinic today, or that are being used in some of the approved vaccines, in terms of how they interact biophysically and biochemically, that make them challenging to think about stabilizing for a vaccine product that might require a lower cold chain storage requirement. And as Neil was saying, thinking about strategies for how do we reduce the number of doses. So it's this interesting combination of us doing biological engineering of the molecules and the hosts that are making these products, but also in trying to advance at the same time our understanding of the manufacturing strategy itself, and improving on that element as well. And so it's been a live fire test of a lot of the ideas that we've really been thinking about as a lab over the last 5 to 10 years, as we've looked at different aspects of the manufacturing and seen this really play out now on the world's scale. And, I think just what I wanted to end with before, we spend some time to listen to some of the questions you may have, is to just point to where we think we want to go now, or where we are going. I think one of the things that we've really come to appreciate as we've looked at vaccines globally is that this is probably one of the largest ways we can address socioeconomic inequities in kind of socioeconomic and in gender equity globally. And this is not obvious at first. If you think, historically vaccines are used for treating humans to prevent disease, and there's clear benefits of that. But another category is in livestock, and livestock vaccines have to be an order of magnitude cheaper and made at an order of magnitude larger volumes in order to actually cover and prevent diseases in those populations. Livestock are sources of capital, wealth for many small shareholders, and are also very important for the kind of daily life of small shareholders in different parts of the world. Most of the world still farms through that type of agricultural enterprise. How do we get these kinds of medicines at the level and scale? So it's going to take different kinds of innovations than what we've been able to do. And so thinking about this COVID problem is one that, the world is going to find solutions to this, and we're excited that we're contributing to this, but there's another stage and another challenge that lays in front of us that we're super excited about going after next, that we think could have real meaningful impact as we look ahead into the next couple of decades. I will stop here, and I think we'd be excited to take any of your questions that you guys may have for us. I think there's a few of them that are starting to roll in, so I think we'll just go through those now as we have time. So we have a question, just kind of like, how are we making the vaccine for COVID? And it looks like we're just kind of taking some of the DNA and putting it into something to produce it. And absolutely, right? At the end of the day, we're making a recombinant protein. So this is done by taking that DNA and putting it into a host cell to produce it. But the nuances of doing that so that we can make several kilograms of this type of product is challenging. And some of the things that we're learning about the molecule itself, and kind of highlighted here on the screen, there's little patches that are more hydrophobic. This leads to interactions of those molecules that leads to reduced stability, which means that they won't last in a cold chain for as long, or at room temperature for as long. So there's elements like that we're starting to target and be able to go in kind of surgically with high precision to engineer out. And as we do that, we can increase the kind of volumetric production that we get from those, as shown sort of in the bar graph that's presented here. So there's a question about, do we think yeast is equally conducive to producing mRNA vaccines as it would be for, say, recombinant viral vaccines? I think the source of mRNA that can be used to manufacture can be made in a lot of different systems, microbial systems like yeast or bacteria. It can also be made by chemical synthesis. So there's a number of different strategies for the mRNA vaccines that are out there. We're particularly excited about yeast for proteins because they can be grown at very large volumes. So it's possible to go to reactors of, say, 50,000 liters, or even ultimately to reactors that are maybe even 150,000 liters. This is an enormous scale. And so if we start thinking about the need for billions of inexpensive doses, these are still some of the best technologies for scaling up to that volume for production. Neil, do you want to take a question?
Sure. I see a great question about if the yeast system will be conducive to adopting the vaccine to new variants. And that is definitely true in this case. We have, I'll answer it in two ways. We're making a version of the vaccine that is kind of the smallest possible protein that you could think about making a vaccine from. So because of that, it is actually more susceptible to the vaccine evolving around it because there's less of it. But on the flip side, the yeast system is so rapid to generate new proteins and test them that we can kind of keep up with the new variants and make them in 2 weeks and then test a new version of the vaccine immediately. Or you could think about manufacturing many different variants all at once and kind of create what's called a multivalent vaccine that could have protection against multiple variants.
There's a question here about the recent reports out about the current vaccines and the potential that they show less effectiveness against some of the new variants, particularly the South African one that's in circulation. I think this is a point of concern globally. Everyone is talking about it. I was just on a meeting yesterday with a number of folks from the Gates Foundation funded network to talk about aspects of this. And Neil's already in the lab making these variants, and we've already tested it with some of the animals that have received our vaccine candidates to see how they may have responded. So far we're seeing actually really nice data from those results. I think the question in vaccines is really what degree of protection are they affording, and the way that it gets handled in the news, it's often yes or no, but it's a question of severity of disease. What is it protecting against? And we've seen this a bit played out in the discussion of the different vaccines that are currently under consideration or are approved as to what stage of disease do they actually help against. I do think there's concerns about this new variant. It has certain changes in the receptor binding domain, the kind of small part of the protein that's shown here, that are worrisome with respect to evasion from some of the responses that individuals have had already to the virus naturally. And so the question now becomes, what about the vaccine? So we'll have to see. I don't know that there's enough data yet to address that, but I think it also highlights what Neil was saying, the speed with which we can adapt these types of structures. We're already looking at new strains to send to vaccine manufacturers today, making those new variants. And so I think part of the strategy going forward will be what's the right way to manage and think about these variants. This is the natural evolution of a pandemic disease, though, as it adapts into the host to understand how best to survive. Great question here about whether we're looking at continuous manufacturing processes. Absolutely. So one of the advantages of some of the new innovations in manufacturing technologies in the biopharmaceutical space over the last few years is taking what's been effectively very large lab experiments. Today, most manufacturing processes of these types of things, you run a batch of fluid with cells in it, you isolate that fluid and you do something else to it, you take it somewhere else on the facility and you do something else to it. You can imagine that's a lot of touch points. Continuous flow of processes through that allow you to intensify or reduce down the footprint required to be able to make that same volume of material and with fewer touch points. Some of the automated systems that we have been talking about or developing in the lab over the last few years for kind of end-to-end production take advantage of some of those continuous operating principles to allow for connected operations. And we've recently spun out a public benefit company, Sunflower, that is taking those technologies forward.
I see two questions I can answer kind of together. One is how did we get a vaccine so quickly this time? And two is, can we develop routines for proactive vaccine preparation? So how can we think about readying ourselves for the next pandemic when it comes? And I'll say that there were two parts that were necessary for the speed and that can be improved still for the next time. One is what we're talking about here, which is rapid vaccine development and manufacturing. So going from a product sequence to something that can be tested really quickly is an important part of it. And then setting up manufacturing processes for that kind of at risk before we've done a clinical trial and know that it's going to work, we're already manufacturing it. That's one part. The other part that often is overlooked and I think is important is that people knew about coronaviruses before this pandemic came. And so back in February, when we and many, many other groups were looking at what to actually make, it was known and based on homology to viruses like SARS and MERS, which obviously weren't the same scale of pandemic, but the fundamental research that went on about those viruses was absolutely critical, such that people knew what proteins could be effective vaccines. So both of those sides are important for future pandemics, both manufacturing readiness and more improved concepts like continuous manufacturing and distributed manufacturing such that we can respond rapidly, but then also like fundamental virology research is so critical to be able to understand the pathogen the moment we see it.
And I want to pick up on one point that Neil just made there about manufacturing readiness. Today, if we wanted to make the same vaccine at multiple sites in the world, it would require taking that process that we've decided, the steps to make that, and figuring out how to fit that into an existing facility in places. One facility might be able to operate that process with a certain set of variations in it. Another one might have a different set of variations. And the challenges are those the same variations and is it the same product? Standardizing and thinking about modular manufacturing facilities like the kinds of automated systems we've been developing is a strategy for how we could be ready in the future to be able to facilitate that kind of transfer. You know, imagine instead of having to ship a yeast strain on an airplane that can't fly, as we found out because of the restrictions on travel, that you could download sequences to places where they could then make their own yeast strain according to the, you know, the blueprint for doing so and operating it in the same kind of facility. We do this with 3D printers and other technologies that exist today. Even the computers that we sit on, you know, they have the ability to perform the same operations in different places. This doesn't exist as a concept generally in manufacturing of biopharmaceuticals today, and is something that we think is exciting as a future direction. All right, so you know, one question that's come up is thinking about the 2-dose vaccine versus 1-dose vaccines. Is one better than the other? I think, you know, this is where the clinical data is really important to help establish what is an effective dose, what's the right dose for that vaccine. Each vaccine that's developed will have different characteristics. When we think about low middle income countries and providing access to medicines there and vaccines like this one, there's a number of features that go into what we would call our target product profile for that. We would like it to be 1 dose. It's much easier to distribute that way. We would like it to have reduced cold chain requirements. And so these are features that we try to design in as we go through this iterative process of vaccine candidate development. So yeah, great question. Great question.
Chris, I'm going to jump in here because I think we need to give our guests time to jump to another breakout room. But thank you so much to both Chris and Neil. I hope that you all found this enlightening and interesting and stimulating, and it's just been an absolute honor to watch the work that they're doing. So thank you so much. Feel free to jump to another session if you'd like, and we'll be back again in just a few minutes.